So today is class 1, and in today's class we're going to discuss about the introduction part. We're going to discuss about the basic morphology, the functioning of kidney, and about the nephron also we're going to discuss. So before proceeding, let's discuss about some core features of Auricle. So as we know, in the Auricle, we provide live classes, which enhance the learning of the students, and also in live classes, there will be direct interaction of the faculty with the students. And then also in Auricle, recording sessions are there in which the student have the access of all the subjects anytime, anywhere through the recording sessions. Then also, Oracle provides ultimate comprehensive book with CBME based question answers, MCQ, and full zone for self assessments. Then also at Oracle we provide MCQs that are from beginner level to expert level, which contain standard multiple choice questions. The students can also book a slot for personalized counseling session in which there will be the direct interaction of the student with the counselor. Then also at Oracle, we provide plans. So we have three plans, basically the basic, standard, and the premium one. So in basic, it is of 3-month plan, and the services which includes in basic plan, it is of pre-recorded classes, live classes, quick revision examination module, and it's cost around 15,000. Then in standard, plan which is of 6 months, it costs around 30,000, and the services are included pre-recorded classes, live class, live doubt clarification, quick revision examination module. Then in premium plan which is of 12 months, it is cost around 50,000, and the services includes our pre-recorded classes, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module. So the premium plan, which is of 12 months, includes all the services which provided by the Oracle. So I would recommend all the student to join or to take premium plan, right? And also for 20% instant discount, you can use my code, that is Aanchal 20. So as we have started the series of renal physiology, so today on 9th March, we are having class on introduction to renal physiology, and these are the classes which going to be happen on these various days. So, talking about the excretory system first, means what is renal physiology? So, as we know, the excretion is a process by which the unwanted substances and metabolic waste are eliminated out from the body. And these waste are like urea, uric acid, water content, right? So, the excretory system plays a vital role in maintaining homeostasis and serve many function, which of the one is the excretion of waste, right? And also it produces and conserve vital ones, that also we're going to learn. And in this topic we'll be discussing the role of GFR in urine and formation and factors affecting it. So this is the clinical case scenario that we're going to discuss in the end of today's session. So as we can see, this is the cross-sectional view of kidney and the diagram of nephron. Yes, so as we know that a large amount of waste, carbon dioxide are produced in tissue during metabolic process, right? And in addition, residue of undigested food, heavy metals, some drugs, some toxic substances, and also pathogenic bacteria or organism like bacteria are also present in our body. So all these substances must be removed to keep the body in a healthy state, right? So all the waste product which is not needed by the body should always eliminated out of the body. So we have various organs or we have various system in the body that performing those excretory function, right? So the one is the renal system, right? The second is the digestive system. The third is the lungs. Fourth is skin. And fifth is liver. So as we know, the renal system the kidney forms urine. That's the way the urine get eliminated out of the body. Then in the last unit also, in last system also, we talk about digestive system. The products which is not absorbed by the body get eliminated out through the anus. Then in lungs, there is removal of carbon dioxide and water vapors also. And through skin, it excretes water, salts, and some other waste also. And also it removed heat out of the body. Then, as we know, the liver excretes many functions like bile pigments, heavy metals, drugs, toxins through bile. So these are the system which are responsible for the excretory function, right? And in our series, we're going to discuss about the renal system, right? So basically, in renal system, we discuss in renal system, we're going to discuss about a pair of kidney, then ureters, then urethra. So as we know that kidney produces urine, right? And ureters are those structures which transport the urine into the urinary bladder, right? And urinary bladder is voided from the, basically urine is voided from the bladder through the urethra. So we have, in renal system, we have a pair of kidney, ureters, urethra, and the fourth one is urinary bladder. Right? Now, one by one, we're going to discuss about the major function of kidney. So, as we know, the primary function of the kidney is homeostasis, right? And it is accomplished by the formation of urine. So, what happens during the formation of urines? Kidney regulate various activity in the body, which is concerned with the homeostasis also. And we're going to learn about those activity. So, in those activity, first we have the excretion of waste products, right? So as we know, in the excretion of waste products, so kidney excretes the unwanted waste products like urea, uric acid, creatinine, bilirubin, right? So first we have urea. So urea is the end product of amino acid metabolism. Then second we have uric acid. And this uric acid is the end product of nucleic acid metabolism. Then third we have creatinine. And this is the end product of metabolism in muscles, right? Then we have bilirubin. So this is the end product of hemoglobin degradation, right? And also kidney excretes many harmful chemical substances also, like some toxins, some drugs, some heavy metals, and pesticides also. Okay, so the first function we have excretion of waste product. Okay, then another function we have maintenance of water balance. Right? So the kidney maintains the water balance in the body by conserving more and more water. Okay. And when it decrease and excreted water, it is excess in body. Right? And this is the important process for homeostasis also. So as we know that the kidney has the structure and function unit that is known as nephron, and various structures are present which are responsible for the conservation of water inside the inside the kidney. Then third we have maintenance of electrolyte balance. Now we're going to learn that how the electrolyte get balanced inside the body. So maintenance of electrolyte balance, especially sodium, okay, especially sodium in relation to water balance, we're going to learn. So what happens? Kidney retains sodium if the osmolarity of body water decreases. So means always kidney start retaining sodium when the osmolarity of body water decreases, and eliminate sodium when this osmolarity increases. Means when the solutes inside the body or the electrolytes in the body are present in very excess amounts, so that time kidney starts eliminating sodium, and when they are present in very less or in decreased amount, that time it retains. Okay, so the third function is maintenance of electrolyte balance. Then fourth we have maintenance of acid-base balance. And for the maintenance of acid-base balance, we're going to learn this thing in very detail. Okay, so the pH of blood and body fluids, as we know, should always be maintained in a narrow range, right? And for the maintenance of acid-base balance, we have other maintaining systems also, like buffer system, then we have lungs, then the strongest system we have the kidney. Okay, so as we know that body is constantly under the threat to develop the condition of acidosis, right? Because a lot of acid produced during metabolic activities. So as we know, various metabolic activities that occurs inside the body, so that's why the body always remain in the threat of develop the condition of acidosis, right? So this is prevented by various organs also, right? As in blood, it is maintained by the buffer system, then also lungs, then also we have kidney. And among all of them, like buffer system, lungs, kidney, the kidney is the strongest one. Okay, so the kidney play a very important or a major role in preventing this condition, that is of acidosis. And kidney are the only organ which are capable of eliminating certain metabolic acids like sulfuric acid, phosphoric acid, so that the acid-base balance inside the body remain maintained. Okay, and this topic again I'm repeating, we're going to discuss in detail, right? Then another function we have hemopoietic function. So as the name suggests, hemopoietic function means, as we know, that kidney stimulates the production of erythrocyte. Okay, and how? By producing a hormone that is known as erythropoietin. Okay, so erythropoietin is the hormone which stimulates bone marrow to produce more and more number of RBC. So that is how the kidney also produces this homopoietic function. And along with erythropoietin, it also secretes a very important another hormone that is known as thrombopoietin. And the thrombopoietin is the hormone which is responsible for the production of thrombocytes or for the production of platelets. Okay, so this is all about the homopoietic function. Then six, we have the endocrine function. So the kidney secretes many hormone. Okay, as we have discussed, erythropoietin, thrombopoietin, then we have 125 dihydroxyvitamin D3, then we have renin, then we have prostaglandin. So as the kidney produces these hormone also, so it also participates in the endocrine, right? Then another function that the kidney plays a very important role in regulation, long-term regulation of arterial blood pressure also, by regulating the volume of ECF and through renin-angiotensin mechanism also. And renin-angiotensin mechanism we're going to discuss in detail in the next class. Okay. Then another thing is that kidney plays a role in the regulation of blood calcium level also. How? By, as we know that kidney secretes this 125 dihydroxy vitamin D3, right? And this is the activator. Basically, this is the activator of vitamin D. And vitamin D is very necessary for absorption of calcium from intestine. So this is how the kidney also plays a very important role in regulation of blood calcium level. So this is all about the major function of kidney, and this is the very important function. These all are the very important function. And another thing, this is a very important question for the university exam also. So again, I'm going to repeat the major function of kidney. The first we have regulation of blood fluid osmolarity and volume, that we have discussed. Then electrolyte balance we have discussed, and acid base balance we have discussed. Then blood pressure, as we have discussed, it is responsible for maintaining long term regulation, right, of arterial BP. Then excretion of metabolic products, foreign substances, then excess substances like water. Then also it helps, it also produces the secretion of some hormones, that is erythropoietin 125 dihydroxy, vitamin D3, then renin and prostaglandin. So these are the major and important function of the kidney, right? Then next we're going to discuss about the functional anatomy of kidney. Okay, so as we know that kidney is a compound tubular gland, right? And it is covered by connective tissue capsules, right? So this is a compound tubular gland. It is covered by connective tissue, right? And there is a depression on the medial border of kidney also, that is known as hilum, through which the renal artery, renal pelvis, and some nerves and ureter passes, right? Now we're going to learn about the different layers that is present in the kidney. So different layers of kidney. We have first is outer cortex. Then second we have inner medulla. Then third we have renal sinus, right? So as we know, this outer cortex, right? So as the name suggests, outer means it is the outer part, right? So this cortex is a dark appearance granule, and it contains some structures like renal corpuscles, then convoluted tubule, and at interval, the cortical tissue penetrates the medulla also in the form of some columns, and that columns are known as columns of Bertin. Okay, so this is all about the outer cortex, the outer structure of the outer layer of the kidney. Then second, we have inner one and the medulla. Okay, so medulla contains some structures like vascular structure that are arranged in parallel radial lines. Okay, and then also the base of the some pyramids are there. Okay, that are known as Malphigian pyramids, and the base of each pyramid is in contact with cortex, and the apex projects into minor calyx. Okay, so about those structures also we're going to discuss in detail. Then next we have sinus. Okay, so in sinus, the upper expanded part of ureter called renal pelvis. Then there are some subdivision of pelvis, in which it includes 2 or 3 major calyxes and 8 minor one. Then third, in renal sinuses, we have some branches of nerves, arteries, and tributaries of veins. So this is all about the different layers that is present inside the kidney. Okay. Now talk about the blood supply to kidney, right? So for the blood supply, we should know some detail about some structures, some veins and some artery. So this is the renal medulla. Then this is the cortex part. Then we have interlobular artery and vein. Then we have interlobar artery and vein. Then we have segmental. Then we have renal artery and vein. And we have arcuate artery and vein. So basically, by these veins and artery, we going to learn the blood supply of kidney. So as we know, the heart supplies direct blood to the kidney, right? So basically, when there is a ejection of blood from the left ventricle to the aorta, then aorta supplies direct blood to kidney, right, through the renal artery. Then through renal artery give branches or drains into segmental artery. From segmental artery, the blood will drain into interlobal artery. Then from lobar artery, the blood will drain into arcuate artery. Then from arcuate artery, the blood will drain into interlobular artery. And finally, from lobular artery, the blood will drain into afferent arteriole. And from this afferent arteriole, the blood will drain into the nephrons, right? So the glomerulus, and then from glomerulus, the blood will exit through the efferent arteriole to the peritubular capillaries or to the vasa recta, right? Then peritubular capillaries will drain into interlobular vein. Also, the vasa recta drain into interlobular and arcuate vein. Then from arcuate vein, the blood will drain into interlobar vein. And again from interlobal vein, the blood will get inside the renal vein. And from renal vein, the kidney direct supply the blood back to the heart through the inferior vena cava, and inferior vena cava drain blood into the right artery. So this is how the blood supply to the kidney occurs. So the intralobular artery, afferent arteriole, glomerulus, from efferent arteriole to capillary network surrounding the tubular system of nephron, and interlobular veins are the collecting vessels of the nephron capillary system. So as we can see, when the blood comes from the heart, all are the arteries which are responsible for carrying the blood to the nephron. Then from nephron through the efferent arteriole, it will exit, and all are the veins which are responsible to carry blood back to the heart again, right? Now we're going to discuss about another important structure, that is nephron, right? So as we know, the nephron is the structural and functional unit of kidney. So nephron is the structural and functional unit of kidney, right? So in each kidney, there are about 1 to 1.3 million nephrons are present. Okay. Then the number of nephrons start decreasing after about 45 to 50 year of age. Okay. Then let's talk about some parts of kidney. So basically, the nephron has two important parts. The one we have renal corpuscles, and second we have renal tubules, right? So we're going to learn about both of these parts through a flow chart, right? which will be very helpful to all the students. So, when we talk about the parts, so as we know, it has two important parts. The first we have renal corpuscles. Then second we have tubular portion. Right? So, in this renal corpuscle, we have two important structure. The first is known as glomerulus and second is known as Bowman's capsule. And about both of these, we're going to learn in detail. Okay, so the renal corpuscles has two parts, the glomerulus and the second has Bowman's capsule. Then the tubular portion has again I'm going to draw. The tubular portion has PCT, loop of Henle and DCT. Okay, so this PCT stands for proximal convoluted tubule. This LOH stands for Looper Henle and this DCT stands for distal convoluted tubule. Okay, so this PCT also has two parts. The first we have, not PCT. So this loop of Henle has two segments basically. The first we have descending limb and second we have ascending limb. And both descending and ascending limb has thick and thin part. It also has thin ascending limb and thick ascending limb. Now about all the structures we're going to learn in detail. Okay. So as we can see in the picture also, the nephron has this DCT part. It has descending limb, it has ascending limb, it has collecting tubule, it has PCT, right? So we're going to learn about all these parts in detail. So as we have discussed, nephron is the structural and functional unit of kidney. Each kidney is made up of about 1 million nephron and each nephron has two parts. We have discussed the renal corpuscle part and the tubule part, right? So the first we have the glomerulus. So what is this glomerulus? So basically it is a tuft of capillary, right? Which is enclosed inside the Bowman's capsule. So if this is the Bowman's capsule and here we have the afferent arteriole, right? Here we have afferent arteriole. This is Bowman's capsule. So basically, the glomerulus is a tuft of capillaries enclosed by Bowman's capsule. So it consists of glomerular capillaries interposed between efferent and afferent, right? So this is the glomerulus we have, and here the exit through the efferent. Right? So basically, this is the vascular system in the glomerulus, which is purely arterial, right? As we know that glomerular capillaries arise from the afferent arteriole, right? And after entering in the Bowman's capsule, this afferent arteriole gives many large capillary, right? And these capillaries are arranged in irregular loop. As we can see, irregular loops are present, right? And also they form the anastomosis, right? Now, all these capillary finally unite to form the efferent arteriole, right? But the diameter of this efferent arteriole, always remember, the diameter of this efferent arteriole is always smaller than the afferent one. Okay, the diameter is always smaller than the efferent one. Now, I hope all the students are very much clear about this glomerulus, right? One more thing is there, that glomerulus capillaries, that basically these capillaries are made up of single endothelial cell. Okay, single layer of endothelial cells which are attached to the basement membrane also. Okay, and these endothelial cells has a very important structure basically that is known as fenestrated pores, or we also call it filtration pores. Okay, so these endothelial cells, basically the glomerulus is composed of single layer of endothelial cells, and these endothelial cells has fenestrated or filtration pores. So always remember, right? Now another thing we have, the Bowman's capsule. Okay, so now the Bowman's capsule, as we have seen, it is the capsular structure, right, which enclose the glomerulus. As we have seen, this is the glomerulus, right, and this is the Bowman's capsule we have. Now it is also formed of two layers. The first is the inner visceral layer, and the second is the outer parietal layer. So as the name suggests, the inner one is the visceral and the outer one is the parietal. Parietal. Now, this layer covers the glomerulus capillary, right? And it is also continuous with this parietal layer at the visceral pore, and parietal layer is continuous with the tubular portion. From here, we're going to start with the tubular portion. So this parietal layer is continuous with the tubular portion of the nephron, right? So this is all about the Bowman's capsule. Basically, the structure which encloses the Bowman's capsule is the structure which encloses the glomerulus. It has two layers, the inner, the visceral, and the outer, the parietal layer. Inner visceral layer covers this glomerular capillaries, and this parietal layer is continuous with the tubular structure, right? Now, next we have the another structure, that is tubular portion. Right. So as we have discussed already, in tubular portion, we study about PCT, DCT, and loop of Henle. Right. So first we have PCT. So the PCT is the portion, basically this is the coil portion, which is arising from this Bowman's capsule, right? This is like this. This is the coil portion arising from Bowman's capsule, and it is situated in the cortex parts. Okay. Now it is continuous with the descending limb of loop of Henle, right? And the length of PCT around 14, the length of PCT is around 14 mm and the diameter is 55 mu. Okay, so this here we have PCT, this is Bowman's capsule, and here we have descending limb of loop of Henle, and this is ascending limb. Okay, now now this PCT also, this PCT also is formed by single layer, sorry for the interruption. PCT also formed by single layer of cuboidal epithelial cells. Okay, so this is also composed of single layer of epithelial cells. Now, the characteristic feature of these cells, these epithelial cells, the characteristic feature is that they also give hair-like projection. Okay, so they also give hair-like projection which is directed towards the lumen, and because of the presence of these projection, the epithelial cells here known as brush border cells. Okay, because of these projection here, these epithelial cells are known as brush border cells. Right? So this is all about the PCT. Now another thing we have loop of Henle, another structure we have loop of Henle. Right? So as we know, it has two parts, the descending part and the ascending part. Right? Now, this descending part has two segments, the thick segments and thin segments. Right? Now, this thick segment is the direct continuation with the PCT. As we have seen, this is Bowman's capsule, this is PCT, then we have DCT, sorry, then we have descending limb, then ascending limb, then we have this DCT, right? So this thick descending segment is the direct continuation with the PCT. So here we have thick, then it become thin, and it descends down into the medulla, and it is length of about 6 mm and diameter is again of 55 mu, right? And it is formed of brush border. It is formed of brush bordered cuboidal epithelial cells. Okay. And this thin one is formed of flattened epithelial cells. Okay. Clear? So basically this thin part is continuous with this thick part. Okay. And this thick part has brush border cuboidal epithelial cells, but it has only flattened epithelial. Okay. Now again, the ascending also has the thin part and the thick part, right? So talking about the thin ascending limb first. So this thin ascending segment is the continuation of hair a hairpin band also, and again it has flattened epithelial cells, and again this thick part also has brush bordered cuboidal epithelial cells. Right? So this is all about the loop of Henle. Now the next we have DCT. Right? So this is DCT is the continuation with thick ascending segment. As we have seen, if this is PCT, then we have descending, then ascending, then here we have DCT. So this DCT part is the continuation of ascending thick segment, right, of loop of Henle, right? Then again, it is in continuation with here. It is in continuation with collecting duct or collecting tubule. Okay. Right. Now the length of this portion, length of DCT is approximately 14.5 to 15 mm. Okay. This is the length, and the diameter is 22 mu. 20 to 50 mu. Okay. Now, talking about the functional histology. So basically, this is also composed of single layer of cuboidal epithelial cells. Right? Now, one more thing, that these epithelial cells are also known as, always remember, intercalated cells or the eye cells. This is the basic morphology. This is all about the basic morphology of DCT. Okay. Now, finally have the last structure, that is collecting tubule, or we can call it collecting duct. Right. So this collecting tubule, as we know, it is the continuation with the DCT. Right. Now, the lower part of the duct lies in medulla, and 7 to 10 initial collecting ducts unite to form the straight collecting duct, right, which passes through the medulla. Okay. Now, the length of the collecting duct range between 22 mm. Then the diameter is 40 to 200 mu, right? Again, it is also formed of cuboidal or columnar epithelial cells, right? And the collecting duct has another two type of cells. The first is the principal cell, which has a very significant function that we're going to learn in further classes. And second, again, it also has intercollecting cells like DCT, which is also known as eye cells, right? So this is all about the morphology of the collecting duct. Okay, then these are the main processes that occurs inside the nephron. First is filtration, then reabsorption, then secretion, excretion. About these steps also we're going to learn in detail in the class of urine formation, right? So basically, filtration means whenever the filtrate passes from the glomerulus to the DCT, then to loop of Henle. Reabsorption means the reabsorption of the substances which is required by the body, then those substances get reabsorbed from the filtrate. Secretion means the basically those substances which does not required by the body that get back or secreted inside the filtrate again. Then the excretion is the excretion of those substances which is not required by the body, like water, urea, uric acid, creatinine. Okay, so this is all about the function of the nephron. Now we're going to discuss about the types of nephron, which is again a very important one, important question. So we're going to learn up the difference between cortical nephron and the juxtamedullary nephron. So before starting with this structure, I would like to draw a diagram to explain the students basically how these structures looks, right? So as we know, the this kidney has two parts, right? The cortex part and the medulla. The outer is the cortex and the inner is the medulla, right? So basically, this cortical nephron, these arise from this part. Okay, so the Bowman's capsule located in the cortical region, whereas loop of Henle, the some part of loop of Henle is innervates or present in the medulla part. Okay, now the second, the juxtamedullary nephron. So again, it started some part of the Bowman's capsule located in cortical region of the kidney, and the loop of Henle located in the medulla part only. Okay. Now, again, as you can notice, the juxtamedullary nephron has long loop of Henle and this cortical nephron has short loop of Henle. Okay. So this is cortical nephron and this is juxtamedullary nephron. Okay. This is the outer part, which is known as cortex, and this is the inner part, which is known as medulla. Right? So this is the structure. I have drawn the structure to make the basic understanding for the students. Now we're going to learn the difference. Okay, now starting with the difference. So the percentage, talking about the percentage. So basically, this cortical nephron, the percentage is 85%, whereas juxtamedullary percentage is 15%. Okay. Then talking about the situation where the renal corpuscle is situated. So as we have seen this, the renal corpuscle of cortical nephron is situated in the outer cortex near the periphery, as we have seen, right? And of the juxtamedulla is located in inner cortex near medulla, right? Now, talking about the location of loop of Henle, as we have seen, basically the loop of Henle in cortical nephron is short and the juxtamedullary, it is long, right? And it is, it penetrates only up to outer zone of medulla, right? And in juxtamedullary nephron, as we have seen, that it penetrates up to tip of papilla. Now the another thing and the very important function or the very important difference about the function. What is the difference between the function of cortical nephron and what is the function of juxtamedullary nephron? So the function of the cortical nephron is formation of urine. Okay. Whereas the function of juxtamedullary nephron is mainly concentration and also formation of urine. So this is all about the difference between cortical nephron and juxtamedullary nephron. And again, I'm repeating to the students that this is the important question, and several times it has asked in the university exams also, right? So this is all about the difference between cortical nephron and the juxtamedullary nephron, right? Now the function. So as we know, as we have discussed the filtration. So this is the first step of the urine formation. Also, this is the bulk transport of fluid uh, from blood to kidney tubule, and then also the result of hydrostatic pressure. As we know, the rate of GFR is 180 liter per day, or if we talk about in ml, so it is 125 ml per minute, right? GFR is again a very important topic, and we're going to learn in detail in the class of urine formation, right? Then another function we have reabsorption. So this is the process of retaining or of returning filtered material to bloodstream, and 99 of what is filtered, right? So basically 99% of thing get reabsorbed back to the blood. Then it mainly involve transport proteins. Normally glucose is totally reabsorbed back into the blood. Then we have secretion. So basically the material which is not required by the body, it get return back to the filtrate, right? So material added to lumen of kidney from blood, active transport usually of toxins and foreign substances. So basically these are not required by the body. So get back added to the filtrate, and through filtrate, the filtrate get becomes the urine. So basically these substances, these toxins and foreign substances get eliminated out of the body, right? Then we have excretion. So basically, excretion is the loss of fluid from body in form of urine. So basically, how we calculate the amount of solute excreted is it equals to amount of filtered plus secreted minus whatever amount is reabsorbed. So basically, as we know, the 99% is reabsorbed back. So this is how we calculate the amount of solute that is excreted, or basically the amount of urine that is going to excreted out of the body, right? So in summary, we have that kidney play a very important or vital role in securing homeostasis, and the nephron is the functional unit, and the processes that involved is the tubular in urine formation is GFR, tubular reabsorption, tubular secretion, excretion. Then juxtaglomerular apparatus monitors sodium and electrolyte load in macula densa and regulates renin release in response. So about juxtaglomerular apparatus, we're going to discuss in detail in next class. Then angiotensin is also there, which is an important hormone that causes vasoconstriction. Aldosterone and ADH release stimulates thrust, raise BP, and secure homeostasis by maintaining GFR, right? Now, the clinical scenario case we're going to learn that. So, a 32-year-old male present with persistent hypertension. So, on examination, his blood pressure is 160 by 95 mmHg. Further investigation revealed elevated renin levels. Which component of the JG apparatus is likely dysfunction? So, here are the options we have. You have 30 seconds to answer. Yes, the answer is C, juxtaglomerular cells. So I hope the students find this class useful. So thank you for watching my class.